Okay, so welcome everyone to the Division of Rheumatology Grand Rounds, our last Grand Rounds of the academic year. So thank you for joining us. Just a few notes before we get started. Dr. Ormseth is going to save about 10 to 15 minutes at the end of her lecture to answer questions. So please feel free to type those into the Q&A function. You're welcome to type them in throughout the lecture and she'll get to them at the end. And also, if anyone would prefer to ask their question over audio, you can feel free to use the raise hand function and I can promote you so that you are able to ask your question. And now I am going to pass things off to Dr. Elkhan.
Okay, hello. So yes, this is the last seminar of the academic year, and we're pleased to have Michelle Ormseth from Vanderbilt, Tennessee, give us an informal seminar on microRNAs and rheumatoid arthritis. She tells us that we in Seattle should not wilt at these temperatures. That's commonplace for her, so we'll try to stick it out a couple of days more. Anyway, just by way of history, she trained as a fellow at VU and was appointed as assistant professor about 5 years ago. Her research interests include vascular disease as well as inflammatory markers in rheumatoid arthritis and lupus. And we look forward to hearing your seminar.
Great. Well, thanks a lot for inviting me to speak virtually with you all today. And the title of this talk is Human and Microbial Small RNAs in Rheumatic Diseases. So I'll send greetings from Vanderbilt. This is our hospital here. My office at Vanderbilt is located just beneath the vantage point of this picture. And I was telling Dr. Elkhan, I was in Seattle maybe almost 10 years ago, and it was one of my favorite trips. It was one of those rheumatology research workshops. And my husband came with me and we stayed afterwards and went on a whale watching trip and had a blast. And it was cool even in June. So I feel sorry that this is going on now for you. And I think I don't know if any of y'all have been to Nashville, Tennessee before, but it's a really interesting place too. A lot of people think of it as more of a, a country music place. And you can see over here, this is our downtown honky tonks. And we have country music, we have all kinds of other music too. And that's starting up again, even in light of COVID. But my family and I don't tend to go down here too much. We tend to enjoy going on beautiful hikes. And this is my daughter and my husband. So if y'all are ever in Nashville, please let us know and we'll show you some beautiful sights. The outline of this talk will be a discussion of what microRNAs and other small RNAs are, their clinical use with an emphasis on rheumatoid arthritis and lupus, and then a discussion of microbial small RNAs in rheumatoid arthritis. And I suspect that most of the people I'm talking to are rheumatologists and researchers in the area who really need no prompting and understanding why rheumatic diseases are important, but there may be some others in the audience, perhaps some rotating residents, med students, or others who may not be as familiar with our diseases. So for the sake of those folks, rheumatologists, please bear with me for a few slides. So people tend to think in rheumatology, we deal with rare conditions, and we do, and we love to treat them. But overall, about 1 in 12 women and 1 in 20 men will develop an inflammatory autoimmune disease in their lifetime. So these overall are pretty common and we need to know about them. And our most common rheumatic disease is rheumatoid arthritis. Patients with RA have about 15% higher prevalence of some functional disability that starts really early in the disease course and then can persist, particularly if we have poor disease control. And even if we ignore the pain that these patients experience and the impact on quality of life, the patients also experience increased cardiovascular morbidity which you can see here, can approach that of diabetes, particularly, again, among those with poor disease control. And so the increased cardiovascular morbidity and increased other comorbidities like cancers can also increase risk of mortality, which you can see here. And despite all this, we still lack understanding of the cause of rheumatoid arthritis and a lot of other rheumatic diseases. For RA, at least, this slide depicts some of the things that we know that occur. And although rheumatoid arthritis is a systemic disease affecting the lungs, eyes, blood vessels, and other organs, I'm concentrating here on the synovial joint. In a normal joint, there's this beautiful, thin, cellophane-like synovial membrane that surrounds the joint and makes synovial fluid to lubricate the joint. And in the setting of RA, this becomes like a thickened mass looking like cooked egg whites. And within this, there are these fibroblasts-like synoviocytes that proliferate wildly and have impaired apoptosis like a tumor. We call this a pannus. As part of this pannus, there's an infiltration of immune cells. And in RA, we find that in the circulation and in the joint, there's a paucity of these T regulatory cells and an excess of Th17s, which are thought to promote autoimmunity in RA and set up this chronic inflammatory response within the joint. There's also an excess of pro-inflammatory macrophages, which pump out the pro-inflammatory cytokines like TNF-alpha and IL-6, which are key targets for the treatment of RA. And then the pannus affects other local cells, causing cartilage damage and secondary osteoarthritis, and also an imbalance of bone-forming osteoblasts and bone-eating osteoclasts, which all can promote erosions. And we're still working out how all of this occurs, but recently folks have found that dysregulation of small RNAs like microRNAs can contribute at all of these steps in the pathogenesis of rheumatoid arthritis and the propagation of its inflammation and tissue damage. So what exactly are microRNAs? Well, this is a picture of the Cathedral of Junk in Austin, Texas. We in Nashville consider this a lesser music city, but until the last several decades, people thought that these short pieces of RNA were just junk. And then investigators started to notice that there were certain genes that just created very short transcripts that could downregulate genes through RNA-RNA interaction. And these seem to be interacting with the 3' untranslated region of other messenger RNAs. And we now know that the microRNAs were binding through a complementary seed region, which is located in the 5' end of the microRNA. So when the microRNA binds through its seed region with the messenger RNA's 3' untranslated region, within what we call an RNA-induced silencing complex, usually causes degradation of the messenger RNA or a block in translation. This leads to lower levels of the protein. One microRNA can regulate many different genes, and oftentimes these genes are within the same pathway, and multiple microRNAs can regulate the same gene, and as such, they work in synergy to fine-tune gene expression. And microRNAs are just the tip of the iceberg, though. This is a figure from a review article written by one of my collaborators, Casey Vickers. And here Casey has shown that there's all these kinds of long RNAs, and then there are these small RNAs. And we think of long RNAs, these messenger RNAs, long non-coding RNAs, and the like. Well, long non-coding RNAs and ribosomal RNAs, for example, can be cleaved into shorter segments of RNA, which can be then classified as small RNAs and may regulate genes in a different manner. And then there are other small RNAs present, like tRNAs, which, as we remember, bring in amino acids to help produce proteins, but they're also cleaved into halves or fragments, which we call tRNA-derived RNAs or TDRs. And these can then regulate like microRNAs or use alternative methods of gene regulation. And many of these can be detected in the plasma or in cells. Now, not all of the small RNAs which a cell produces are used within this cell of origin. Rather, some are exported out of the cells within structures like exosomes here, which are very widely studied. And also, they can be exported out in things like lipoproteins. And I'd mentioned my colleague before, Casey Vickers, and he actually discovered that high density lipoprotein, or HDL, can take up small RNAs from one cell and deliver them with functionality to other cells. And so we can study small RNAs in particular cells of interest, and we can also study these small RNAs within the blood, and they are protected there within these structures like exosomes and lipoproteins. MicroRNAs are being evaluated for the treatment of disease. On the left is one of the initial drug studies looking at an antisense drug to human miR-122 for the treatment of hepatitis C. And on the right is a list of microRNA therapeutics currently in development. For diagnostic and prognostic use, a 10-member panel of microRNAs shown here is used in conjunction with an oncogene panel and currently FDA approved for classifying indeterminate thyroid nodules. You can see here that those patients in green who have a low risk panel result have a much greater probability of not having a malignancy, whereas those with a high risk panel result are much more likely to have a malignancy. And understandably, this can have a lot of important relevance to taking care of the patients at bedside. Similarly, we started looking at human plasma small RNAs simply to discriminate between RA and control subjects. To do this, we started with a discovery cohort of 165 patients with RA and 91 controls who were frequency matched for age, race, and sex. From plasma, we extracted RNA and did next generation sequencing on the small RNA pieces. And then we used differential expression and a machine learning method each to choose the top 15 candidates that could best discriminate. And the machine learning component that we use just helps to make our results more generalizable and limit the influence of patient subsets on the results. And we remeasured these candidate microRNAs in the same discovery cohort samples by PCR for several reasons. One, sequencing has some biases, so it's always best to validate findings by a different method. And two, for downstream use, it's much more simple and less expensive to use PCR. Then we use some additional machine learning methods to reduce the number of small RNAs to the smallest, most parsimonious model that best distinguish between RA and controls, and then tested that model within an external validation cohort of 32 patients with RA and 32 control subjects. And we identified this panel up here with 6 microRNAs such as miR-22-3P and others, all of which were enriched in rheumatoid arthritis, except for this one here, 96-5P, which was decreased. And we found that in both the discovery and validation cohorts, the plasma small RNAs together had a high area under the curve to distinguish between RA and control subjects. And this plots the 1- specificity and the sensitivity of the test. A perfect test would be 100% sensitive and specific, making it a perfect box shape. whereas a test that was no better than a coin flip would fall around here where the red line is. So you can see that the performance was very good among both discovery and validation cohorts, but most importantly, it had very similar performance in those with seropositive or seronegative RA, and regardless of whether the patient was in drug-free remission or highly active, or regardless of drug they were using. So we're currently working with Michael Hollers, which some of you likely know, at University of Colorado, and with one of the Navy bases to use the DoD serum repository to determine if these microRNAs or perhaps other small RNAs might help predict those who are at risk for RA who will go on to develop RA. And we've been looking at circulating small RNAs for use as markers or predictors of important comorbidities and outcomes in patients with rheumatoid arthritis. One extremely important comorbidity in patients with RA is cardiovascular disease, which, as I mentioned earlier, is increased in patients with RA and one of the biggest contributors to early mortality. And this is something that my mentor, Mike Steen, has focused a lot on in his work, how to detect those with greatest risk and find novel ways to reduce it. And he, along with my colleague and good friend, Cecilia Chung, brought this to the attention that really the risk factors that are used in the general population underestimate the risk patients with RA have. And here Cecilia showed that in patients with RA, they have a greater probability of having coronary atherosclerosis than control subjects across any given Framingham risk score. So we tested whether plasma small RNAs could help improve classification of cardiovascular risk in patients with RA. And in this case, to help classify those with actionable high coronary calcium scores. I mentioned earlier, that kind of the methods that we use using some unbiased machine learning methods. And we use these same methods to develop a 9-member microRNA panel featured here. And we found that this panel improved the discrimination of those patients with high atherosclerosis burden beyond clinical predictors like age, sex, ACC, AHA 10-year risk score, DAS28 score, in which we use the SED rate, which in our hands we feel is better than CRP for predicting cardiovascular risk in RA, and also presence of diabetes. And although this panel was discovered by unbiased methods, we found that by pathway analysis, most of the panel members were predicted to or known to affect pathways leading to atherosclerosis. In addition to accelerated atherosclerosis leading to increased risk of MI, patients with RA also have increased heart failure independent of atherosclerosis. So I worked with some of our cardiology colleagues at Vanderbilt on this paper looking at incidence of heart failure in patients with RA. And here we have nearly 10,000 RA and 10,000 control subjects, each with about a median of 8 years follow-up. And you can see that the cumulative incidence of heart failure in RA in red here is increased compared to that of controls. It was about nearly 5 versus nearly 4 per 1,000 patient years in the raw unadjusted analysis. And still after adjustment for all these major covariates, there was still a 21% higher incidence of heart failure in RA. And then among those subjects who developed heart failure, both RA and these controls, you can see that among the patients with RA in red, they had nearly 70% higher mortality compared to the non-RA controls here in blue. So clearly we have a lot of interest in predicting heart failure in RA. And we suspect that microRNAs may be helpful to us to do this and are planning future studies in this. But for now, I just wanted to show you some data in the general population. So our incoming division of cardiovascular, the incoming director of the Division of Cardiovascular Medicine at Vanderbilt is going to be Jane Friedman, and she and her group used the Framingham Offspring cohort, including over 2,000 subjects without heart failure or abnormal echoes, to determine if plasma small RNAs are associated with incident heart failure. And she and her group identified these 3 microRNAs that had a significant inverse relationship with incident heart failure. All had very similar relationship, but here we show miR20A as an example. And you can see that for every 2-fold increase in the microRNA, there was a 15% reduction in incident heart failure, independent of known risk factors. And again, these were discovered using unbiased methods, but each of these 3 microRNAs had significant prior animal model data and in vitro model data demonstrating their role in heart failure. So in vitro studies implicate a different microRNA, which may be important in lupus. miR22-3P, which can affect many expression of many different genes which are relevant to lupus. And this is a volcano plot that demonstrates differential expression of plasma microRNAs in lupus versus matched control subjects. And this is actually just a small study, a pilot with 12 lupus and 12 control subjects. Each dot represents 1 plasma microRNA. Those that are more to the right are more enriched in lupus. Those more to the left are more enriched in controls. And you can see that there are many microRNAs which tend to be enriched in lupus. This one here, which is circled, is miR22-3P. And it caught our eye because not only was it clearly enriched, it was also the most abundant microRNA in lupus plasma. So, you know, even a doubling of it is going to have a massive effect. And we were able to validate that this particular microRNA was quite increased in patients with lupus doing another modality, PCR. And to see what the role of miR22-3P in lupus was, I had the opportunity to work with Amy Major, who's also in my division of rheumatology, and works with the SLE123 mouse model of lupus. And in her lab, folks gave this lupus-prone mouse IV injections of a miR22 blocker or a scramble control, which shouldn't change the level of miR22. And the mice received the injections every 2 weeks, starting just prior to disease onset for 10 weeks total. And you can see that using the miR22 blocker, we were able to reduce miR22 in the splenic T cells as well as B cells as expected. And Amy and her lab found that by blocking miR22-3P, they were able to reduce the splenomegaly in these animals and the lymphadenopathy compared to scramble control. And it also decreased anti-double-stranded DNA antibody titers and nephritis scores. On the top panel shows the pathology from the scramble control treated animals. So these should have the normal level of miR22, and that looks similar to the untreated animals. And on the bottom, you can see the results of blocking miR22 with decreased infiltration, decreased cellularity at the glomeruli by H&E, and decreased immunoglobulins on immunofluorescence. And although we don't know which specific targets miR22 was affecting, Amy's data suggested that miR22-3P regulated T cell differentiation, and blocking it led to a reduction in a subset of T cells that produce a lot of interferon gamma, which led to this improvement in disease. So this is a proof of concept that a microRNA detected in the plasma may have clinical relevance for understanding the biology of disease and potentially discovering therapeutics. Another small RNA class, which could be important, are tRNA-derived RNAs, or TDRs, which I'd mentioned before. And as you recall, the tRNA can be cleaved into halves or fragments. And this is another volcano plot showing differential expression of TDRs in lupus versus control plasma. And this one here circled is a TDR derived from a tRNA which encodes glycine. And I worked with my colleague Casey Vickers, who I mentioned, and his postdoc Danny and Michelle, who found that this TDR was able to downregulate ROCK2, possibly through its perfect C region binding with ROCK2's 3' untranslated region. And many of you know ROCK2 is important for determining the balance between regulatory T cells and Th17s, which is important to lupus, rheumatoid arthritis, and other rheumatic diseases. And Danny found that the TDR decreased Th17 skewing and expression of IL-17 and 21. So in this case, we find that a TDR discovered in plasma has what appears to be a homeostatic role in lupus to dampen excess ROCK2. And this is really interesting because ROCK2 is an important target of interest in lupus and other autoimmune diseases. There's currently a clinical trial going on for using an oral ROCK inhibitor for scleroderma, and I suspect lupus will be coming soon. So we and others have looked at microRNAs and other small RNA classes in the plasma, like these listed here, tRNA-derived RNAs, yRNA-derived RNAs, small nuclear, small nucleolar, ribosomal, and others. And we, even after permitting mismatches to the human genome, we still find that there are a lot of these unmapped sequences here in gray, which we can observe from sequencing results. And folks started to wonder if those could possibly be of non-human origin. And this is one of the earliest papers to look at this in human plasma. They evaluated plasma from 3 healthy subjects, 3 with colorectal cancer, and 3 with ulcerative colitis. In each disease state, the results were averaged, and then they used different strategies, 3 different strategies here, with increasing number of mismatches to the human genome. And so in each of these disease states, in strategy 2, they've permitted 2 mismatches, which is actually a lot given that these are short RNAs in the range of about 16 to 40 nucleotides in length, but still there's a lot of sequences which match perfectly to bacterial genomes and fungal genomes here in red and green. They also found that these small RNAs were resistant to treatment with RNases, meaning that they were likely in some sort of a protective structure. One protective structure may be bacterial extracellular vesicles, or in gram-negative bacteria, these are called outer membrane vesicles. And this is a schematic of a gram-negative bacterial cell wall. And just as humans produce exosomes, so too bacteria can produce vesicles, like outer membrane vesicles, which contain small RNAs. And just as exosomes or HGL can traffic small RNAs between cells, these OMVs can as well. And so how do I know that they can affect human cells or maybe even human immune function? Well, one example comes from a group who is focused on why Pseudomonas was able to colonize patients with cystic fibrosis. They were looking at OMV virulence factors, which promote the survival of Pseudomonas, and found that OMVs were loaded with these small RNAs, typically tRNA-derived RNAs and ribosomal RNA-derived RNAs. And when they incubated the OMVs with the human bronchial epithelial cells, the OMVs were able to transfer their contents to the epithelial cells, which was evident in increased counts of small RNAs that aligned to Pseudomonas when they sequenced the human bronchial epithelial cells. And then they also chose several very abundant small RNAs which were present in the OMVs and followed those and found that those specifically were able to be transferred to the human bronchial epithelial cells just after exposure to the OMVs. And they evaluated the function of one tRNA fragment that was very abundant in the OMVs. And in a series of in vitro and animal model studies, they found that while the OMVs were studded with LPS, which should cause inflammation and neutrophil recruitment to the area, the OMVs delivered this TDR to the human bronchial epithelial cells, which downregulated all of the MAP kinases in a signaling pathway. And that led to decreased IL-8 expression, decreased neutrophil recruitment to the lung, and facilitated chronic infection. So in this case, a TDR was also acting as a virulence factor to help Pseudomonas evade host immune response. And so we wanted to know if microbial small RNAs were altered in patients with rheumatoid arthritis, because we know there's a lot of microbiome disturbances in rheumatoid arthritis. And because based on the study I just mentioned and quite a few others that are building now, the microbial small RNAs may actually reduce host immune responses, which could be a good thing in patients with RA. So to answer this, we sequenced small RNAs from plasma and used what we call the TIGER pipeline. And this was developed by Kwon Hoo-shin, who is in bioinformatics, and he goes by Tiger, and this of course was named after him. And the pipeline will first align all of the small RNA sequence reads to the human genome with up to one mismatch. And then there are a pool of what we consider small RNAs that may be potentially microbial in origin. These are then aligned to representative genomes of the human microbiome, environmental bacterial genomes, fungal genomes, and then we added a RA-specific module that included genomes of bacteria which have some relevance to RA based on literature, whether that be by microbiome study, by culture data, antibodies, what have you. And in order to be counted as microbial in origin, the small RNA had to match perfectly to these genomes. And at this point, we can use genome counts to compare, for example, genome counts of E. coli in RA versus controls. One of the problems here, though, is that these, as I mentioned, are typically tRNA-derived RNAs and ribosomal RNA-derived RNAs that have a lot of conservation across many different species. And so it's actually quite challenging to know for sure which bug contributed to that small RNA count. But we can do our best. But we do feel much more confident that if the small RNA maps perfectly to these genomes, it may likely then be microbial in origin, and we can look at these individual sequences as though they were just like other microRNAs, like miR22-3P, and we can compare them across diseases and disease states. One concern I had initially was that perhaps these microbial small RNAs are actually just contaminants. And anyone who does microbiome studies will say that each kit has its own microbiome. So in all of our studies, we run no template controls through the whole process from RNA extraction to sequencing. And here, while there are some microbial small RNAs in this showing here, there's also human small RNAs in the no RNA control as well, but it's very few compared to what we observe in plasma. And these are bar graphs where each bar represents one individual subject in the study. And we found that overall about 20% of plasma small RNAs map perfectly to microbial genomes, you can see here in green, in both RA and control subjects. And while they were pretty similar across these diseases, we do see there's considerable variability across subjects. And on the right, you can see that in looking at the makeup of the microbial small RNAs, the majority found in plasma mapped to the phylum Proteobacteria, which is here in green. And this is similar in RA and control subjects. And thinking about what source these microbial small RNAs may come from, we can look at the phylum level microbiome composition at various sites in the human body, as shown here on the left. Here also we can see Proteobacteria in green, and it's a bit more common in the oral cavity. And also in the stomach, particularly if there's an H. pylori infection, because H. pylori is from the phylum Proteobacteria. But a limitation of most microbiome studies, particularly those examining gut microbiome by stool, is that it's examining luminal contents rather than what may be most intimately associated with the host. And to my knowledge, the area in the human body that has the greatest proportion of Proteobacteria in microbiome studies is within the gut mucosa, particularly deep into the lamina propria, where these bacteria can come in close contact with host immune cells and even translocate. So these bacteria may be particularly relevant to disease. And in our study, we found that there were about 10 microbial genome counts that were significantly decreased in RA. And these come from gram-positives, gram-negatives from a variety of phyla. One particularly interesting one on this list, to me at least, is Lactobacillus salivarius, which was decreased in our study in RA. And this is interesting because a prior microbiome study found that Lactobacillus salivarius was actually increased in saliva, plaque, and stool of patients with RA. And in this particular study, we didn't assess stool or oral microbiome in these individuals, so we don't know if they inversely correlate. But like I mentioned before, because of the conservation of tRNA and ribosomal RNA sequences, it's hard to know for sure with confidence that these are the particular microbes which contributed to this difference that we saw. We do have much more confidence in looking at the sequences. And this is another volcano plot from one of the modules where each dot is a small RNA sequence, a microbial small RNA sequence, I should specify. And you can see that the composition of these sequences is altered in RA. And we concentrated here on these three, which were enriched significantly in patients who have rheumatoid arthritis compared to matched control subjects. And these are the altered sequences here listed from the volcano plot. Each of these is about twofold increased in RA significant after adjustment for age, race, sex, batch, and multiple comparisons, of course. And what's interesting is they appear to be the same base sequence, just progressively longer. And we found that these weren't just an artifact of sequencing, confirmed that TDR1 and TDR3 here are both enriched in RA. We didn't measure TDR2 because it's so closely similar. It would be very hard to isolate that one by PCR. The top three microbial small RNAs which were enriched in RA all come from the same tRNA, which encodes for arginine. And tRNAs are produced in a very reproducible manner. They're cut close to the anticodon loop to produce halves, like we had here with TDR3. And they're also cut in the D and T loops reproducibly that produce these shorter fragments, which were what we consider TDR1 and TDR2. The bacteria that produce these are gram-negative aerobes from the phylum Proteobacteria, and there's lots of different microbes that can produce this particular tRNA. So we want to see if these were in any way related with RA disease activity or clinical features. And this is a heat map showing the positive associations in red and negative associations in blue. And on the bottom, there are microbial small RNA features listed, which were either the total microbial small RNAs and other features which were significantly altered in RA. And then on the right are clinical features in RA, importantly, tender joint count, disease activity score, and the swollen joint count. You can see that these measures like TDR1 are inversely associated with these clinical features, particularly tender joint count and the other swollen joint count. And we found that this relationship remained regardless of adjustment for certain medication use. But we know that medications can affect microbiome and gut permeability. So we thought maybe possible that this was just something related to medications that the patients were using or how well we were able to control their disease. So to test this, we examined whether the microbial small RNAs were modified by drug treatment. We used plasma samples from a multi-center study called TETRAD, which was led by Lou Bridges and Jeff Curtis. And none of these sites were Vanderbilt, so patients wouldn't overlap. I'm not sure if you all were part of this study or not. I'd have to go back and look. But the patients here had blood drawn before and 6 months after their physician started a new drug as part of standard of care. And in this case, they started either methotrexate, adalimumab, or tocilizumab. And then we had 6-month follow-up data and blood samples from the patients. We didn't find that there was any significant change overall in the microbial read counts before or after treatment, and the composition of the microbial small RNAs didn't change either. And these are histograms just showing change. So everything here hovers around a change of 0 for total microbial small RNAs, plasma TDR1 and TDR3. And this was similar across when we looked at individual drugs as well. And what was more interesting though was that among those who improved on treatment, they had 3-fold higher total abundance of microbial small RNAs, and increase in the TDR1 and increase in TDR3, showing that these are associated actually with better treatment outcomes. And when we looked at the disease association with microbial small RNA features, we see now that in both independent cohorts, there was this persisting inverse association between the same microbial small RNA features and joint counts, with the strongest being this association with TDR1 and actually tender joint counts. And so we wanted to know what the function of these microbial small RNAs may be, particularly given prior studies that the microbial small RNAs can affect human immune responses. And Chong Wu, who's a postdoctoral fellow working with me in the lab, has been working on this TDR1 project, and she tested the effect of TDR1 on THP1 transformed macrophages. And the reason for this is that, there's many different cells that we could test that have strong relevance to RA. But my rationale was that, our blockbuster drugs that block cytokines that macrophages produce a lot of, and every drug that's effective in RA reduces macrophages within the synovium. And given that they're part of the innate immune system, we thought that would be pretty important also if we're thinking that the small RNA may be a virulence factor. In any case, Chung found that delivering TDR1 to the macrophages compared to a scramble control or other microbial small RNA or no RNA control resulted in a really significant reduction in expression of a number of genes, which we measure by a nanostring panel. And Chung validated them as well by PCR and also in repeated experiments. And you can see the list of transcripts which were reduced. And you can probably, people here that know a lot more about immunology than I do can look at this list and see a common theme that these are primarily interferon response genes. And we still, of course, perform pathway analysis, and based on the reactome pathways also showed here that, you know, TDR1 really reduced type 1 interferon signaling. And this is really interesting because some studies have shown that type 1 interferon is associated with more tender joints in RA, possibly due to direct stimulation of the nerves. And this is why we get, you know, when we get viral infections, we have a lot of body pain. Studies also have shown that higher type 1 interferon is associated with poor response to therapy in RA. TDR1, which seems to decrease type 1 interferon response, is associated with fewer tender joints and better response to therapy. So kind of our associations kind of hold true with our clinical study here. And we really think of type 1 interferon as being associated with viral infections and lupus, but these are increased in RA and other inflammatory autoimmune rheumatic diseases as well, so showing a common theme. So potentially TDR1 could be an important marker or an important mediator in other inflammatory autoimmune diseases. But I don't think that one particular microbial small RNA concentrations that we observe in plasma is controlling RA, but I think that together these may be important effectors, just as we see with human small RNAs. So we have a lot more work to do on TDR1 to find out what that and other microbial small RNAs are doing in rheumatoid arthritis, as well as lupus and other rheumatic diseases. So in summary, we just showed that circulating small RNAs are powerful markers of disease with therapeutic potential, that microRNAs are just the tip of the iceberg when it comes to small RNAs, and that circulating microbial small RNAs may be a link between the microbiota and human disease. And so I'd like to thank a lot of folks. It's lovely working here because I work with a lot of people I really like, and I've featured Chung here. She's been working on the microbial TDR1 project and a lot of other things that are in the pipeline that are just too green to share today. Casey Vickers and Amy Major, who contributed a lot. And then my mentor here, who always hides from pictures, who's kindly worked with me since I was a rheumatology fellow. And so I'd like to stop and answer some questions in the chat if there are any. I'm going to stop share so I can see what's going on.
Okay. So while people are sending you questions, and if you don't see them, I can probably pick them up here. Maybe I'll start. Yeah, I'm really intrigued by these tRNA changes, cleavages. So I'm sort of interested in how that happens and whether inflammation influences cleavage of the tRNAs.
It really does. So there's one, for example, angiogenin is what cleaves the tRNAs into halves. So it cleaves in that anticodon loop. And in the setting of inflammation, angiogenin is increased quite a bit. And so, and that corresponds with an increase in the tRNAs as a class that you can observe in the cells and also in the plasma. And so I think that in part that could actually be a homeostatic, supposed to have a homeostatic effect, because for human tRNA fragments, we often see them as similar to microRNAs, kind of dampening these inflammatory responses. But there's other things that they can do. It's quite interesting. There's a TDR fragment that they found in that's increased in hepatocellular carcinoma. And its function is that it actually binds to the messenger RNA that encodes for ribosomal proteins. And so it doesn't act like a microRNA, but in that messenger RNA, it actually has a little hairpin loop, and it can bind in that hairpin and loosen the hairpin and increase the translation to the ribosomal protein. And that promotes more cell division and the hepatocarcinoma itself. And so when you block that tRNA fragment, you can decrease the proliferation of the cells. So not all of them are have that homeostatic role, but it is more common to see.
And I think, if I remember right, there's some old literature from Paul Plott saying that some tRNAs are chemokines. Have you come across that?
I haven't. tRNAs is the without being cleaved, just as the whole origin. I didn't know.
Okay. Okay. There's a question here from Thomas Mustel, and he asks you, do you think microbial RNAs evolved as a tool for microbe immune symbiosis?
Yeah, I think it could be, so there's more, if you think about it in RA, this is probably a good thing. If it actually does decrease host immune response, that could actually be quite nice. But from the bug's perspective, I think it'd be advantageous to decrease host immune response. There, and this isn't just in humans and bacteria either. There was a beautiful science paper that I just didn't have time to share with you all, but this was I believe it's a tomato plant, some kind of a tomato plant, and it gets infected with a fungus that actually transfers RNAs to the tomato plant for its own survival. So, but that can actually harm the tomato plant. But I think maybe in humans, there'd be more an idea of symbiosis. The other thing I found in RA is that this TDR was increased among, particularly even higher among patients who had a longer disease duration. And I wonder if there was something about that they were good enough to get in our study, healthy enough to be in our study and things like that. So it's a possibility. Oh, I see another one. Can your methodology also analyze somewhat larger RNAs? So what we end up doing is when we prepare the sequencing libraries, we size select the things that should be about 40 nucleotides in length and shorter. And it's a bigger segment, but because how we have the barcoding on the ends of the RNAs, but we've actually size selected a bit smaller, because if we include, for example, everything that's 200 nucleotides in length or smaller, or included, say, the messenger RNAs and long non-coding RNAs that aren't, cut, that would consume all of the sequencing reagent, and we wouldn't be able to see it because they're longer. So that's just to help us see those small RNAs better.
So presumably, if we're talking about methodology, you also enrich for small RNAs, or if you're working with, I guess, lipid particles, you do some enrichment. Is that right? Or?
So yeah, so the way that it works is we add these adapters and barcodes to the end of the small RNAs. So because of the chemistry of the small RNAs, at least the ones that we detect, we don't see the messenger RNAs because they won't be tagged because of, for example, the poly A tail. Yeah.
Okay.
And then for lipids, we haven't, we're still working on some of that data, like HDL, small RNA data. But yeah, we do in some of our studies working with Casey Vickers, we isolate out HDL, purify it, and then sequence the small RNAs from the purified HDL. Yeah.
Good. Okay, in the last minute, so you mentioned, I think, the reduction in rare 1720A, I think it was, in RA or heart failure. So, I mean, any ideas on the biology there?
Oh, the 20A, yeah. that was actually in the general population rather than RA just yet, and that actually works on TGF beta. Ah. Yeah, isn't that interesting? all three of those ones that they identified affect TGF beta, which are quite lovely, you know.
Makes sense. Okay. Well, I think we are at the end of, we're coming to 10 o'clock now. So I do want to thank you very much for this terrific lecture. Lots of things to think about. And I know you'll be meeting some of the faculty today and over the week. And you're on call, you're attending right now, aren't you?
Yeah, yeah. Medicine attending at the VA.
Medicine attending, wow. That's a sacrifice. So thank you very much. We appreciate it.
Okay, great.
Okay.
Thanks a lot.
Bye-bye.
Appreciate it.